This is a paper that changed my life. It put me into contact with Peter Molenaar and Connor Dolan. We worked on multivariate modeling every Thursday night in Peter's office, and it really shaped my research for the rest of my career. We first would go out for fries and then do some very heavy statistical modeling afterwards. And Peter was a great mentor without any sort of feeling for why people might not be following what he was telling me. So we had a problem in time series modeling, and one day Peter phoned to my house and said, "I've solved the problem." And my husband said, "Dorret is not here." And he said, "Oh, that's not a problem. I'll just explain it to you. It involves modeling a two-pleach matrix, and if you can give the message to her, I'm sure it'll be fine." So I want to talk a bit about multivariate modeling and the seminal contribution of this paper, and also try to address one other point, and that is, it has been repeatedly said this morning that we should all read the old papers, but the old papers are really sort of inaccessible. And I'll try to give a few hints of what you need to know in order to be able to access the old papers. So since the 1920s, 1940s, there was this question around. There are very many, maybe an infinite number of phenotypes that nearly all are to some degree influenced by genes. The information in the genome is probably limited. So are the same genes influencing multiple traits? And as Lindon said about this, if we can apply multivariate genetic analyses, we will achieve an increased understanding of how genes and of course also the environment works. In addition, there's a practical consideration. Multivariate modeling will quite often also provide us with increased power. So a paper that was the very first one to address multivariate genetic modeling was published in 1943, but went very unnoticed by the genetics community. It summarizes the situation that phenotypic correlations can arise for multiple reasons, because traits are influenced by the same set of environmental factors, or because traits are influenced by the same set of genes. Distinguishing between pleiotropic effects of genes, a single gene influencing multiple traits, genetic correlations that arise because of linkage, and genetic correlations that arise because of non-random mating. So here's the very first path diagram spelling out that a correlation between traits called X can arise because they are influenced by environments that are correlated, or by genes or sets of genes that are correlated. And you see the well-known decomposition here of a phenotypic correlation into a bit that is due to a genetic correlation between traits, an environmental correlation between traits, and then the importance of the environmental and genetic correlation is weighted by the square root of the heritabilities of those traits. This paper went largely unnoticed. So in the 1960s, 70s, a lot of people were trying to work out how to analyze multiple data from the same person. So for example, Steven Vandenberg, John Lowland, collaborated on this question together, and they came up with a solution that said, If you have identical twin pairs, any differences between them are due to environment. If you have dizygotic twin pairs, any differences between them are due to environment plus the genes that they don't share. So what you could do is create different scores for multiple traits, correlate the different scores for MZ and DZ twins, and then analyze those correlation or covariance matrices. And you can sort of apply any standard techniques for analyzing correlation matrices. It'll give you an impression of the structure of the multivariate structure of the genetics and the environment. Then Lindon started to think about this problem as well, because a sort of multi-step approach where you first compute differences, then look at the correlation between differences, then do a principal component analysis of the different scores. A multiple step approach is obviously not a solution that is ideal, and you want something that does the multivariate model fitting and the genetics in the same step. So this is from a letter written on a train on a Saturday from Lindon to Owen White. And what he does in this letter is really spell out the maximum likelihood solution to analyzing multivariate genetic data. And this is in the notation used, I suppose, by the Birmingham School at that time. So everything that says D, read additive genetics. And everything that says E1, read unique environment. I'll do a bit of explanation later on. But the really important point is here that Lindon refers to the solutions that has been proposed by Karl Jureskoch to do maximum likelihood analysis in a factor analytic setting. So this is what will be familiar to a lot of you, the Jureskoch approach to factor analysis. Simply saying there is a latent construct, and if we talk about individuals, this is a latent score on some sort of factor, and this latent score that we do not observe influences a set of observations. In a genetics context, this factor score could be a breeding value or a genetic factor score. So taking this Jureskoch model, where from the model that I just showed you at the individual level, we can derive the expectation for variance in the population. In the Martin and Eaves paper, there was a decomposition of multivariate data in mono and dizygotic twins based on this particular model. And in the paper, there's an application to cognition data, and there is a second application published shortly afterwards on impulsiveness. And the data analysis took about a year's worth of computer time at the then strongest computer at the University of Manchester. So why is the original paper so difficult to read? You can all sort of immediately understand the factor model when we picture it like this. Well, there are two problems. The model fitting actually was not done on covariance matrices of twins, but was done on matrices of cross products between and within twin pairs. So that's the first sort of difficulty in now reading the paper. And then the second is this sort of different notation that was used in Birmingham at the time. So if we translate the model into sort of literal notation using the path analytic representation, the D stands for additive genetic factors, and the Greek D, delta, stands for the common additive genetic factors, and you can see that those common factors influence a series of phenotypes. H1 stands for unique environment, and H2 stands for shared environment. So that is a first bit of problem out of the way. And then you can, in terms of these common and also specific factors, derive the expectations for the matrices of cross products between and within monozygotic twins. So the old approach used to be the recognition that genetic and environmental variation is partitioned into variance between and within families. And then so the total variance for a trait is of course the variance within plus the variance between families, and those sigmas refer to variance components. And then the other bit you need to know is that somehow the total genetic variance is 1/2 of D, so not all of D, and it is only 1/4 of this particular bit that refers to the dominant genetic variance. As I said, E1 in Lindon's notation refers to within family environment, and E2 refers to what we now call C, the common environment. So this is the well-known biometrical model. The values of the homozygotes are called D, and the value of the heterozygote is called H. Then in addition to P and Q for allele frequencies, the notation U and V is used, but the good news is this all boils down to the better known equation nowadays for the decomposition of genetic variance into allelic effects and allele frequencies in the population. So the other bit is the decomposition of variance between and within families. Clearly, the variance between families needs to be larger than the variance within families if family members resemble each other. And you can get at the between and within family variance based on analysis of variance, or if you're working with multivariate traits on MANOVA. It's a very computationally attractive approach to analyze multivariate data, but we're no longer using it because it also has its limitations. It is an approach that is not very well equipped to analyze sex differences. Analysis of variance works on the assumption that within a family there are no systematic effects of birth order, for example, if you work with twins. It really only works if you have data without any missing values, because the first step is to obtain a mean square or a sum of squares based on the data, and no raw data input can be used. And it applies mainly to data with a continuous distribution, because you cannot model any sort of threshold values. So there were two papers pointing out shortly after the Lindon and Eaves paper was published, one with David Volker and one with Rita Cantor, on how this model can be implemented in Lisrael. And then in the next 20, 30, 40 years, an enormous amount of papers on the application of multivariate genetic models to twin and family data. And maybe it is the greatest honor a paper can achieve that everybody is now so familiar with it that the paper isn't even cited any longer whenever somebody does a multivariate genetic analysis of twin data. And rather than citing Martin and Eaves, people tend to cite Neal and Carden, which is a book that is long out of print. So the major applications have been on the phenotypic side. trying to get an understanding of the structure of multivariate data, but also, for example, can multivariate genetic analyses help in problems of diagnostics? And then, of course, in linkage analysis, there was an application of the multivariate model in order to gain power to find QTLs for continuous traits, and we're now seeing the first applications also in GWAS data. So one extremely famous application of the multivariate model was with Ken Kendler, addressing the questions whether symptoms of anxiety and depression cluster so often because they are influenced by the same set of genes versus the same environmental risk factors. And there was a very clear answer. The comorbidity is because of the genetics, and the distinction is because of the environment. We have been working on an application of the multivariate model to item data from the CBCL in children, addressing the question why those items cluster, and again the clustering into symptoms of depression and anxiety and somatic complaints is clearly because of genes. This is one example of an great increase in statistical power working with data, with longitudinal data on blood pressure. Blood pressure data are fairly stable over time, although notice that the stability is not any larger than, for example, a simple measure that you can obtain from survey data like neuroticism. And using the entire multivariate structure in Dutch and Australian data, we found some interesting hits in a linkage analysis of systolic and diastolic blood pressure. Now, the fact that those models could be implemented initially in Lisrael, later in MX, nowadays also in M+, all sorts of R packages, has led to enormous popularity without maybe sometimes a similar sort of gain in understanding what is going on. And this was also pointed out in 1989, that the ability to apply complex models because of the increase in software and computer possibilities might people sometimes cause to lose control to see the relationship between theory and data or evidence. This is not at all how Lindon approached the problem of multivariate genetics. In his papers, he points out that analyzing multivariate data can give you insight into, for example, the structure of assortative mating, or it can tell you about the distinction between additive and non-additive genetic influences. Also pointed out that if two characters are influenced by exactly the same set of genes, it doesn't follow that you will see a phenotypic correlation between the two characters. In fact, they might be influenced by exactly the same sets of genes, and the correlation between them could just be zero. As explained for the very special case over here, when both characters are influenced by genes that each have 2 alleles per locus, and then depending on the allele frequency and the effect of increasing and decreasing alleles, the correlation could still be zero for 2 traits that are influenced by exactly the same sets of genes. So we should be very careful and indeed go back to the old papers in interpreting genetic correlations or the absence of genetic correlations in making statements about overlapping or non-overlapping genes. So this is my last slide. It's probably going to be said more often during this meeting. A large extent of all the work that we do is because we stand on the shoulder of giants, a quote quite often wrongly attributed to Newton. And I just want to point out that the Free University in Amsterdam is also very privileged to count Lindon Eaves among the Doctor Honoris Causa recipients in the past couple of years.
So it's an enormous honor to be here today, and it's a great pleasure for me. I did not, like so many of the people who've spoken, get to be a student of Lindon's directly. I don't know if you noticed in Ermine's slide, I was sort of on the side there. I was the weird American cousin. But there are a group of us. West Virginia. But, you know, in just thinking about what to say about Lindon, what occurred to me is that I admire Lindon in a very specific and kind of literal way. I mean, of course, I admire him in the ways that we all hear about today, that he's a great scientist and a great mentor and very generous with his knowledge and his data, and he's always fun to be around. But I admire him in a particular literal sense, which is this, that pretty much everything that I, and I think most of us do professionally, think up topics to try and do research on, then try and build theoretical models to analyze those topics and try and find data that we can fit our models to, and then try and do data analysis to come up with parameters from those models, and then write up papers on the basis of those analyses. Lindon is simply the best at every single one of those steps. And I find it's humbling over and over and over again to have to face the fact that I simply can't do the professional things I do as well as he can. And I think that's true for many of us here. And another general Lindon theme, of course, that we've heard even more, I'll return to it at the end, is that every single thing we do in this field, or so it seems to me, Lindon has done before, probably 20 years ago. And that's certainly true of everything I've done. And I'm not going to do one of those nice reviews like several other people have done. I'm just going to talk about one paper in particular that's been a great inspiration to me. It's been up there before. It's this one, A Theory of Developmental Change in Quantitative Phenotypes Applied to Cognitive Development, that he did with Andrew and Jay Long, a name I actually don't know.
Population genetics, I think it's Michigan there? It's not.
And it's going to take me a few slides to get to the actual model that they fit here. But what I want to talk about first is what it is that motivated, in my mind, the model that they wound up fitting. One of the basic motivations to me is to try and find an explanation to probably the second most fundamental finding in the behavior genetics of ability. The first most fundamental, of course, being that it's heritable. But probably the second most familiar finding about the genetics of ability is that the heritability of ability increases with age. Everybody knows that. It's often cited. But I'd like to suggest that most of us, in fact, don't really have a very good understanding of that particular finding because we usually phrase it in exactly that way, about a change in a standardized variance component over time, which is interesting as such, but doesn't really tell us very much about the actual developmental processes that underlie it. And this Eaves et al. Paper is about the developmental processes. And here's one way to bring home to you how little, I think most of us, and this is not true of Lindon, actually know about this finding. If heritability increases with age, ask yourself this, on the simplest level, why does that happen? Does the MZ twin correlation increase with age, or does the DZ twin correlation decrease with age, or both? I mean, that is pretty much the most basic thing you can ask about the finding, and I don't, if you look at the reviews and meta-analyses of the phenomenon, it's not even mentioned. Nobody knows whether heritability goes up because MZ twins are getting more similar or DZ twins are getting less similar. There's another basic, somewhat less recognized finding about the way ability changes with time. And that's that the stability of cognitive ability increases dramatically over time, and particularly as people change from childhood to adulthood. I'm going to make this point with some data that I pulled out of the Dutch twin study. I did the analyses out of the correlation tables in their papers. These are simply the phenotypic time correlations, MZ twins below the diagonal and DZ twins above the diagonal across time. I can't read my own damn slides these days without my glasses, which are all tangled up with the mic, so they're almost big enough. If I turn this way, now I can see them. And unsurprisingly, simply at the individual level, they're very correlated. And we can ask some questions about how they're correlated. One of them, you'd expect the correlations to go down as the interval between the time points increases. So proceeding from the yellows along the diagonal, which are consecutive time points, going out to the blues, which are the farthest apart time points. And yes, they decrease slightly. We'll see data like that again in a minute. We can also ask if the stability increases as the children get older. And maybe a little bit. It's not very dramatic. But I don't think that's the most interesting part of the stability question. The most interesting part of the stability question is the correlation, the abstract variance component way to say it is the correlation between the non-shared environmental components at different time points. And that's the way these were actually computed, though I think it's more useful as we move on to simply think about it as the correlation of MZ twin differences over time, right? That's MZ twin differences are a very rough approximation of the non-shared environmental component. So we're asking if you have a pair of identical twins measured at multiple time points, and you ask which twin is smarter at time 1, how well does that predict which twin is going to be smarter at time 2? And I think the answer in general in childhood is quite surprising. You can find this in multiple data sets. The answer is they're correlated practically not at all. Which is to say that in childhood, identical twins are switching back and forth at random as to which one is smarter as they develop. Now, what's even more interesting about this is to ask the same question in adults. So I grabbed these data from the public part of SATSA, which are 4 measurements, everybody's familiar with it, of adult twins aged 40 to 90, the first principal component of their battery. And here are the same correlations on the top in the adults. So in adults, identical twin differences are correlated almost 1.0 over time. In fact, with a little bit of modeling, that coefficient is often not distinguishable from 1.0. And down below are the exact same correlations in the children. Right? So that is the same parameter in adults and children. You have the exact same parameter, which in adulthood comes very close to being indistinguishable from 1.0, and in the children comes very close to being indistinguishable from 0. That's kind of interesting. Something happens between childhood and adulthood to stabilize within pair differences. And the question is, what might that process be? It's going to bring me back to Lindon's paper. It can't simply be that within pairs they vary randomly, switch back and forth until they turn 21, then stay permanently at the value they reach at 21 for the rest of their lives. Some kind of individual level process has to lead to the stabilization over time. That brings me back to the Eaves, Long, and Heath model. This is it. There's not time to go into all the richness and details of this. But this model, I would argue, is designed in some ways to explicitly to model changes in variance components over time and changes in stability over time. And I'll tell you as another point about Lindon, what I like about this model. It's an interesting, I think, something I didn't realize until I was looking at it the other day. You notice about this model that there are no curved arrows in it anywhere, right? It's all straight lines, which means it's all regressions. And what does that mean to me? What that means is that this is a model specifying and hypothesizing a developmental process at the individual level. It's very easy to make a developmental model of changes in heritability over time. You can fit an ACE model to each of the individual time points, and then fit a variance to each of the A variances and draw covariances between them, and there you go. But when you do that, you're simply describing a covariance structure. This is a hypothesis about how a covariance structure comes about, and that's what makes it powerful. Now, in fact, what I want to show you is some related models that Chris Beam and I have been fitting. And one thing we're working on right now that's quite difficult is the relationship between these models and the Eaves, Long, and Heath models. These models, they also turn out to be related to some of the models that Connor Dolan showed. I'm looking forward to talking to him about it. But these are models that include a particular kind of within-pair gene-environment correlation in them. What they posit is that, in the red arrows, that there's a causal process linking the phenotype at time 1 to the non-shared environment at subsequent time, meaning that the twin with the greater phenotype, the smarter twin at a particular time, then gets systematically exposed to the superior environments down the road. And think about what this model is going to do. It's going to induce gene-environment correlation over time, because the phenotype obviously includes genetic effects in it, particularly in the DZ twins, right? The identical twins, the only thing making them different at time 1 is non-shared environmental or random differences that get transmitted to time 2. In dizygotic twins, there are environmental differences within genetic differences within pairs in the phenotype, which then lead one of the twins to get exposed to better environments at time 2, and those cumulative effects lead to accumulating RGE within pairs over time. Now, what are some of the predictions of this model? The model predicts, and this is based on simulated data, that DZ twins should diverge over time. So it predicts that the answer to the question that I asked at the beginning of does heritability go up because identical twins get more similar or because fraternal twins get less similar, is that the answer ought to be, according to the model, that fraternal twins get less similar, they diverge. Now, to actually look at this in the data, I'm going to go back to the data set that Eaves et al. Analyzed in the original paper in 1986, which is the Louisville twin study. One of the little miracles of Eaves, Long, and Heath is that they conducted their analysis based only on the correlation matrix from those data, which I can't imagine doing. But fortunately, the data themselves are now available, and they are, just for general knowledge of people out there, they're massively underanalyzed. I think there's a lot of work left to be done on the Louisville twin study. It is, I think, the most developmentally intense twin study of ability in history. It has a lot of complexities. This is kind of an odd histogram, but there are up to 16 testings available for each of the twins. About 500, 550 pairs. The other point that it's one of the things we're struggling with right now that this graph makes is that there's, despite all those testings, there's tons and tons and tons of missing data. So there are 75 or so pairs with all 16 time points, and then moving down the scale, lots and lots of twins that are missing them in unsystematic and hard to work out ways. So how do the twin correlations in the Louisville twin study fit with the predictions of our model? Well, the answer is off the bat that they don't. On the left are the MZ and DZ, MZ on top, it's not labeled, MZ on top, DZ on bottom twin correlations from the Louisville twin study. On the right are the slide you've already seen with the predictions from the model. And what you see is that in the actual data, the identical twins get more similar over time as the fraternal twins get less similar. Now, what mechanism might there be to make identical twins get more similar over time? Well, one thing that's not considered here is reliability. These tests are starting very early at 3, 6, and 9 months, and ability tests aren't very reliable at 3, 6, and 9 months. Unfortunately, the tests were not given multiple times to the twins at any single time point, but fortunately, we figured out a way to estimate the reliability of the testings, even though they weren't Twins weren't multiply tested at the same time. I won't take a lot of time with this. What this is this graph shows the correlation between time points separately for each beginning time point. So if we start with that circle right there, the line that follows from that is the correlation of ability at 3 months with ability at all the subsequent time points out to 15 years. And then you can then go into the next one at 6 months and follow that correlation. And here we are at 3 years, and that's the correlation. And then finally, the correlation there at 12 years to 15 years and so forth. And you can see two things. First of all, the correlations go up as kids get older, and of course they decline with interval. Well, there's an available model of how that would happen that we borrowed from Bill Dickens, which suggests that the covariation between tests at 2 time points is an exponential function of the interval in between them. So that if you have 2 tests separated by k years, that the covariance between them is going to be the true score variance that those 2 tests shared by some fixed decay parameter operating over k years, with A is a floor, it's the lowest that that covariance can get. Well, if you fit that to the model there, and then infer what happens at k equals 0 years, when there's no interval between the tests, that's the estimated reliability of the tests. Well, we fit that, the decay parameter and the maximum reliability of the tests are actually confounded with each other. So assuming that, inaccurately, that the tests at age 18 reach perfect reliability, that's the estimated reliability of the tests over time. And it makes pretty good sense that they start between 0.5 and 0.6 and they probably increase to something more like 0.9 than to 1.0. And if you correct then the MZ and DZ twin correlations for the changes in unreliability, you get a very nice match. The MZ twin correlations flatten out and the DZ twin correlations decrease. So correcting for the changes in the reliability in the test over time, you get a very nice fit. I've only got a couple minutes left. The model also predicts the increase in stability with time very nicely. Conclusions. First of all, whatever it is you're doing, Lindon already did it, and you might as well look it up and get that out of the way first. And what I've learned from Lindon is that the goal of developmental models is not to quantify variance components, but to specify causal processes at the individual level. And simply quantifying biometric parameters in time does not do it. And our hypothesis that substantial and increasing genetic components can result from relatively small genetic effects and processes that lead to accumulating RGE. Finally, I'd just like to close with a hypothesis, something we're working on right now. There are 3 basic developmental trends in the heritability cognition, 2 of which I've talked about. It increases with age, it becomes more stable with age. I want to add a third, one that in fact I'm more associated with than the first 2, that the heritability of cognition increases in better environments or gets worse in bad environments. What I'd like to hypothesize is that all 3 of those things are actually the same phenomenon in different forms, that they're all the result of progressive within-pair matching of phenotypes and environments. And in that context, I can offer a definition of what poverty is. Poverty is essentially an environment in which it's not possible for the smarter twin to match him or herself with a superior enriched environment because that environment isn't there to be matched with. And thank you for everything, Lindon. I'll leave you with that thought.
Yeah, you'll notice I have a gazillion affiliations because there's a long-standing, known fact that I'm never where I'm supposed to be. So I figured out I just keep my affiliations active everywhere, and then no matter where I'm at, I'm probably where I'm supposed to be. But I think regardless of where I'm supposed to be in whatever affiliation I have, I am exactly where I'm supposed to be today. I retired from the service a few years ago, and the one thing that we try to do when people retire is bring out all the people you went into basic training with. And so you could think of maybe your PhD program or probably your first academic appointment still kind of like basic training. And so I asked around for some photos of Lindon in his early years, and this is, of course, this picture from Birmingham, which you've already seen. By the way, I had to change my slideshow like 6 times during the previous talks because all the pictures and things I was going to talk about, Nick and others talked about. So you obviously will recognize some of these faces because they've been talked about before with Lindon, Nick, and Lindon's advisor. What you probably started to see in the last few years is things like this in the popular media. Our politics rooted in your genes, the monkey cage is more genes in politics, all sorts of we found the gene for type of literature. And for most of people in the public and most of social science academia, this is their first introduction to the potential that your attitudes, your left-right opinions, your political and social attitudes might be more than what's socially transmitted. And so I just put this little graph together of the number of publications per year on genetic influences on attitudes. And the first one was Eaves and Eysenck in 74, and then the Martin et al. Paper in 86, and then Tom Bouchard and others started picking up in the 90s. And around 2007, this massive jump of interest in the field. And it's this area that towards the end of Lindon's career, we've seen a huge change in another discipline. And so I was going to talk today about that progression of where these pieces are, what they've meant to the field, and what has actually happened outside of behavioral genetics and psychology and in political science, and the profound impact that Lindon's had just in the last couple years. So we'd seen this paper already that Nick talked about a little bit, the 74 paper. And this was quite an advanced paper in many ways. It's a multivariate style type of paper with personality and attitudes. And that was largely because of Hans Eysenck's influence when Lindon was a postdoc with Hans Eysenck. And he conceptualized his traditionalism and radicalism factors, which were mainly attitudes like the death penalty and abortion and so forth, as part of your personality, as some kind of component. But even when they ran, and at this time they had 708 twin pairs, which was quite a lot of twin pairs for in the 70s to get social attitudes on. And they came to the conclusion, although we claim to show some common basis for differences in personality and attitudes, we stress that for the scales in question, the greater part of the genetic and environmental variation is specific to the particular traits. Radicalism scores, for example, are totally independent of the measured personality variables, although they have significant associations with other attitude measures. So they found this genetic influence on both attitudes and personality. They were shared to some degree, but yet both constructs and the influence on both remain unique to themselves. So there's something about attitudes that wasn't being captured in personality. Around this time, this is the earliest picture I could find of myself. My influence in the field obviously wasn't quite there yet. I think I'm 6 in this picture eating a slice of pizza. I was probably around 4 actually when this paper first came out. It took about 12 years later before there was large enough samples to really extend that literature. And this was, Nick Martin and Lindon Eaves and others who worked on this in the Australian and UK data. And you can't see the attitude list there. It's too small, but they had 50 individual attitude items, the heritabilities, and then constructed what was the Wilson-Patterson Conservatism Index. And not only looked at the heritability, but actually brought in the spousal correlations and other relationships to discuss how this transmission is occurring. And Nick talked about that, so there's no reason for me to go into a whole lot of detail. But again, for me, my introduction to the field was still far behind. I just joined the service within a year of that time. Slightly different facial feature then than I have today, of course. But yet no interest in creating anything interesting or genetics that I knew of at the time. A few years after that, the Eaves, Eysenck, and Martin book, 89 book, was bringing all of their research together. And again, Nick talked about this, so I had to shift some things around. It was interesting, what Nick said is that nobody had taken notice. Actually, Nick, I think you're wrong about that, because right around this time is where a lot of psychology started to pick this up, particularly Tom Bouchard and the twins reared apart, using much of the same measures that you guys used. And then Tesser's paper actually used your own data, took the correlation matrices from your published data and put it into his paper. So people were picking it up, just not in the fields of social sciences outside of psychology that focused on political attitudes. And at this time, I still wasn't creating anything. I was interested in jumping out of planes and destroying stuff. So it really wasn't quite part of this field yet. Come the 90s is when Lindon still kept going forward in this research agenda. Although it wasn't well known, he was using extended models, multivariate models, developmental models, exploring the changes in conservatism with age, exploring its relationship with religion, exploring it in much larger extended kinship models with spousal correlations and so forth in children. and contributing to this way, but still wasn't touched or picked up on by the disciplines that most focused on political attitudes, and that was political science at the time. Now, this is where I started to make my transition from the service into finally, as you can see, a very happy face leaving the Army, or so I thought, and beginning my graduate studies. And in graduate school, I started graduate school in political science, actually just walked into the office coming right out of the field, trying to make my transition to be a civilian. And the degree program was only focused on social learning and economic theories. And that is still the main theories in political science today about where you get your attitudes from. It's either directly trained or it's some rational action, and we could care less where it comes from. It's rational behavior. And I found this to be wanting based on all the experience I had, that people just didn't really work that way. And so I came across these two papers, the Eaves and Martin papers, and this was my kind of awakening that something was going on that I didn't know about. Well, I did what all good graduate students do. I got behind my computer, started pounding away and emailing people. I think this is fascinating. I'd really like to work on this data. I emailed everyone in the field who had worked on social attitudes, and almost no response from anyone. And the one response I did kind of get originally was Lindon, who basically told me to bugger off. And the reason being was, at the same time, from the same university that I was training at, Lindon was kind enough to share his data with a few scholars who then said, oh, thanks for this, we'll get back to you. And the New York Times contacted Lindon with a publication that came out on his data without him knowing about it. So not very happy about that time. But this is where Nick comes in, thank goodness. Nick also responded, and he was the one person who responded positively and said, yeah, come on out to Australia, mate, I'll set you up, we'll figure something out, and we'll go forward from there. And so I did. I abandoned my program. I just basically left my university in the middle of my PhD, so that's it, we're done. I will file my PhD here, but I'm not doing any more training there, and I left and went to Australia. And that's when I began to work with Nick very closely, and through Sarah Medlin, her patience, she should be sainted. I had no knowledge of matrix algebra, structural models, genetics, or even basic psychology when I started working with Sarah and Nick. 3 months later, the first publication came out. I largely credit to Sarah's patience dealing with my gazillion questions and driving her crazy. And at that time, that's when Nick said, I really need to send you to Lindon. This is really his area of research. And so Nick kind of smoothed that way for me to begin working with Lindon. And so then I went to Virginia. And this is a real credit to the kind of person Lindon is. You'd heard this before, but the almost drop everything took me from beginning to end to teach me everything that he knew that he could, which of course I didn't learn everything that he knew because half the stuff that you said I couldn't figure out. But I did take enough of it to cause some damage into the field. And at this time, there were maybe 2 or 3 political scientists that would even be remotely interested in this area of research that would think about it. And so Lindon and I began this journey. And this is our typical work day, we didn't go into the office. I went to Lindon's house, into the backyard, and we would spend hours upon hours going through models, questions, ideas, thousands of kind of papers that Lindon had laying around that he started but didn't quite finish. I'd say, Lindon, I had this idea that I think that religion's tied to this. Oh, yeah, I have that right here somewhere. I didn't quite finish this thing. And so we went to finish those off. And during this time also was really my transition to being a civilian and talking about this kind of deep theoretical issues. And one of those issues during this time was this huge gay marriage and homosexual rights debates that were coming out at that time. And so we had decided in one of these meetings after several glasses of wine that Leviticus was really not saying you can't sleep with another man. It says it's just a direction. You can't physically. You have to change your positions because it's an instructional manual. So that was our change. And again, this kind of fits into Lindon's main mode of not being the priest that kind of fits the other mold of other priests. And so a very typical Lindon Eaves kind of moment in that. We also came up with lots of new programs of study to operationalize your sticky bonking theory, to do the love boat experiment with twins. We had all sorts of wonderful ideas in those days. But we did get to work. We got a lot of work done. We published quite a bit of papers, really extending out those earlier works, flushing out what was really going on with this genetic influence of attitudes. And it wasn't just myself. I'm going to talk a little bit about myself in this, but there were a lot of other people involved that Lindon affected through this attitude program. Some of them you'll recognize, Nathan and Sarah, Matt Keller, and of course Brad, who's now at VitBig working with Mike and Lindon. But others that you wouldn't know in this room. In 2010, the National Science Foundation funded a group of political scientists to learn behavior genetics, largely because of the papers that Lindon put out. had a huge impact on this field of political science. And so that spike came from all those faces that were in different disciplines of publications. There's been over 180 publications in the last 4 years in political science and social science journals on genetics and social attitudes. I started listing them and then just gave up. So huge impact. And I think it's not easy to see if you're not in that field, but imagine at the end of your career changing an entire program in another discipline that you're not even related to by the work that you've done in your original field. It's just quite an amazing accomplishment that the folks in this room might not see. Today, now articles featuring genetic methods in political science are in all of our top journals. Almost every issue coming out, there'll be some publication. Funding by the National Science Foundation. In our major organizations like BGA equivalent for political science, the Best Dissertation Award was awarded to a student who did theirs in genetics and political attitudes, which is really quite incredible. And our Emerging Scholars Awards, which is similar to awards in BGA for Best Scholar under 10 years, also someone working on genetics and political science. A huge shift in the field. Opened the door for all sorts of physiological, biological, hormonal, neuroscience publications. It just kind of laid the groundwork. And so this fight that had been going on in social science about behavioralist or rational choices models had just been wiped out with, holy cow, now we've got this whole neurobiological, genetics, and hormonal issue to deal with that might influence human behavior. And so even in spite of this, to give you an idea of the debate, and this is fairly recent, this is one critic said, if genetic influences account for some portion of the variance in political attitudes, it would require nothing less than the revision of our understanding of all human history, much if not most of political science, sociology, anthropology, and psychology as well, perhaps understanding what it means to be human. That is this baseline of behavior and belief in the field. And to see this in the last few years push that out, largely from the work that you had done and then extended by the people that you had trained, is really something else. And so this is just, I was doing a review paper. I was asked to do a review on politics, and I started going through all the publications. And whether it's political knowledge, ideology, authoritarianism, trust, all sorts of different attitude factors, traditionalism, defense, foreign policy attitudes, outgroup attitudes, there is some substantial amount of genetic influence. And then when you get to party identification, which is a lot like which religion you are, then there's really none. And so quite consistent with all of the counterpart literature and psychology doing pro-sociality, emotions, and so forth. If you were to translate this into political attitudes and constructs and behaviors, you'd see roughly the same type of variance components. And so the field had to move from social learning and rational choice theories to, and again you might recognize some of this from Lindon's work, a much broader construction of behavior, DNA being involved, development, structure and function, and of course, your parents met and had sex, which political scientists apparently had never thought about prior to these behavior genetic publications. And this became very interesting to the field. The idea of all the spousal correlations there is, attitudes are amongst the highest, attitudes and church attendance. Stunningly high, actually in Martin data, it's even higher. It's about 0.72 in spousal correlations, which is really phenomenal to think of a correlation between two. It launched a series of papers, not simply because it changed the variance components decomposition when you start including assortative mating, but because of what it would mean phenotypically. Social scientists hadn't really thought about spousal assortation on attitude factors in other domains. And it launched a series of papers looking at dating and mating, evolutionary models, simulations of what it would mean for political polarization, and other topics that you might not even realize this is all going on now, active. Most of these publications are 2012 and 2013 that are forthcoming. And the only reason why they were aware of these high spousal correlations was because behavior geneticists needed to look at them to basically change the parameters in the model and account for spousal similarity and genetic transmission. So that became like this other one area that you would not have seen. The development work you did, and Eric's presentation was really nice on this, talking about these kind of strange relationships, that one of Lindon's samples that he used had co-twin correlations from 9 to 18, asking kids 9 years old what they felt about abortion is kind of interesting. And what they found was between 9.5 and 18, there's really no difference in MZ-DZ correlations whatsoever. You've probably seen this with lots of other traits. But as twins leave home, as they get older, the DZ correlations drop. What was really neat about this contribution of Lindon's, and I was part of this as well, was that we looked at the difference between people who were at home, 18 to 20, and the kids who, and then listed in their survey, this is the adult cohort population, did you still live at home or not? We split those apart. The ones who still lived at home just look just like the 15 and 17-year-olds versus their adult counterparts in the same study. So it's really the home environment was keeping twins more similar. And as soon as that home environment was gone, you saw the DZ correlations drop. And this has launched an enormous discussion. I was just at this special kind of invite roundtable in political science with all of the kind of big sociologists in political science trying to figure out what was going on between when kids left home and when they were young adults. The 74 paper also launched another series of papers, and Brad Verhulst's work is largely based on this, is revisiting Lindon's causality work between personality and politics. And using larger data sets, more robust kind of models, they are distinct and separate. And again, that started to really change the literature in political science, who had always thought personality preceded attitudes, that they weren't distinct constructs. Other work that has really affected the field is policy, really moving not just description, but into public policy, or what we could do with this and what it would mean to the policymakers. An area that you might not see at all if you're focused mainly on description, but a few of us were invited to the DoD to talk about what this would mean for identifying potential violent actors in large populations. Could we use this kind of information? My answer was no, but the fact that they were listening and interested in it was something else, something we hadn't seen before. And so I'm going to end this pretty quickly with two thoughts that Lindon had in attitudes when I first met him. The first one was, if the data are to be believed, this is, you probably, if you ever worked with Lindon, would remember him saying this, the transmission of social attitudes are more than socially transmitted, but the data that we had were limited, and there'll be no individual genes or markers of any large effect. Rather, as Fisher intimated, thousands of genetic influences operating in tandem with development environmental conditions. So there really isn't going to be the gene for liberalism. But yet Fox News disagrees. They say that they actually had found the gene for being liberal. But we decided to put this to the test. I contacted everyone I knew from every registry that had any social attitudes. So we had the US and Australia from 88, 90, 2008, and 1980, Denmark from 2009, Sweden 2009, Hungary in 2011, all sorts of different measures, left-right attitudes, self-report, authoritarianism, egalitarianism, and so forth. And what we found was no matter how it was measured, except for one trait which actually stood out, no matter how it was measured besides this one trait, genetic influences existed and they could be statistically equated. The common and unique environmental influences could not. So the same kind of pattern of individual differences within populations, across populations, despite how attitudes were measured, came out the same. So it looks like we can believe it based on the data. We said, well, can we find the specific polymorphism that might come popping out? Well, with our small sample of about 11,003 populations, no genes, no markers of any large effect, exactly as you suspected. And so I'd like to just leave on a kind of personal note. My career would not be anywhere near where it was had I not begun my work with Nick and Lindon. I was fairly certain after my PhD I'd go back to some type of defense work or something else. And it has been a huge transition in my life to become the academic and person I am today. And so I know for many people it's about the research. For me, it was always about the person who I wanted to be and the things that I wanted to do. And Lindon made that happen. He actually really helped me make that transition to be a civilian. And I'm fairly certain he's had that personal impact on many, many people. And to be able to say that today, that not only wouldn't be the scholar I'd be today, but I wouldn't be the man I'd be today. It was in my time as a postdoc in Richmond that I actually cleared out my weapons locker. I actually retired officially, put my guns away, and that was a huge moment in my life. I was a bit lost at that time. It's a very personal thing to actually explain when you're a soldier for 20 years, what it means to make that kind of transition. And it was my time spent there that did that for me. So more than your academic contribution, your contribution as a human being, I can't quantify or put into a slide. And thank you for who you are and what you've given to me and all the people that I know. And so thank you.
So good afternoon. I was given the task of trying to review the impact of Lindon's work on the field of psychiatric genetics. This, of course, is so intimately tied with my own biography and my own relationship with Lindon that you can see that this was both an abstract but quite personal task. I think the way I'd like to start is to take a broad historical overview about what the field of psychiatric genetics was like prior to Lindon's venture into the field. Modern psychiatry really began in 1917. The great German neuropsychiatrist Emil Kraepelin finally was able to develop his lifelong dream, which was to develop the first modern psychiatric research institute. You see its name in German here. I won't try to butcher it. And it included, among the departments of psychopharmacology, neuropathology, headed by Louis Alzheimer, a department that was the genealogical demographic department, headed by a young psychiatrist by the name of Ernst Rüden. Kraepelin had recruited him because he was then knowledgeable and in fact had mastered the algebra of the laws of Mendel, which were then really hot. That was the new technology of statistical genetics in those days. And in those first years, Rüden and the psychiatric genetics he was performing was at the forefront of the methodological developments in statistical genetics. In particular, Rüden was in close touch with a German pediatrician by the name of Weinberg, who was then solving some of the critical problems of human geneticists. Weinberg was the first to figure out that if you wanted to get the correct recurrence risk for disorders in human families, you couldn't count the proband. So he developed his proband correction method. And Weinberg struggled with the idea that if you have age-dependent penetrance, you really can't count everybody equally as a function of the age they are, and developed the first of what are now a series of more sophisticated age correction methods. So it was this study in 1916 that really sets psychiatric genetics on a scientific course. There had been many case studies and views that psychiatric illness was familiar. That goes back, in fact, to Roman times. But this is really the first time when statistical models of genetic transmission was applied to psychiatric illness and to dementia precox in this case. Over the next 20 years, that is up until, and I'm afraid, into the time of Nazi dominance, and that's a story that we're not going to tell today, the Rüden School kept up and was very vibrant in the development statistically. Bruno Schulz was actually the methodologist of the group. They were had a very clear concept of what it was like to do proper ascertainment. So Luxemburger's first systematic twin study, done in 1934, used a single census day in all the hospitals in Bavaria to count, so they were very concerned about proper ascertainment. And in Rüden's clinic, many of the great next generation of psychiatric geneticists in Europe spent time, amongst them Eric Stromgren, who developed the Stromgren age correction method. There may be a few people in the audience here old enough to remember that, but also Elliot Slater. So Elliot Slater was the mentor of both Irv Gottesman and Ming Shuang, who really were the previous generation. Now, the US, by contrast, was in the dark ages. They were lacking in any high-quality methodology, and especially in quantitative genetics, so that I'm afraid at this same time period in the United States, psychiatric genetics was dominated by what I would call the overzealous Mendelizers like Davenport and the psychiatrist Rosenoff, who had no concept of what, if you've ever read his twin study of schizophrenia, he had no concept of what proper ascertainment would be. His idea was to write all his friends who ran other psychiatric hospitals and say, do you know if you have any twins with schizophrenia, and just list them out and send them to me, and that was his twin study. Any of you that have spent any time reading through Davenport's monographs, this is my most favorite one. I do teach from this, but this is the literal title. The feebly inhibited, the phenotype that he was doing the Mendelian transmission of was nomadism, or the wandering impulse, with special reference to hereditary inheritance and temperament. He developed a single gene model for nomadism as a phenotype. From the middle of the third of the 20th century, England was the world leader in statistical genetics. Many have written about this with more profound knowledge than I. It is true that two strong programs emerged, one based here in Edinburgh and the other in Birmingham. And it is also important to know that each of these two schools eventually reconnected. That is, from somewhere around 1925 to 1930, when statistical genetics and psychiatric genetics were very closely intertwined, there really was this diaspora of 40 or 45 years, especially in the United States, but also in Europe, where the developments really they went their separate ways. Psychiatry kind of closed around. We were much more interested in epidemiologic follow-up studies. The methodology of the genetics that was done was really very simple. Really no progress was made. And the first person really to link up was by then a finishing psychiatric resident by the name of Ted Reich, who was sent from Washington University, St. Louis, really from Sam Guzet, who said, you know, we've been talking about, as Sam and Eli Robbins were, about the importance of family history, how critical this was to the validation of psychiatric illness. And to their immense credit, they said, well, maybe we should really learn something about genetics and send Ted Reich off. And Ted spent 2 years with Douglas Falconer, early 1970s. This was one of the classic papers that Ted wrote when he was still in Edinburgh. Now, when I was visiting Birmingham, and I took part of their MSc course in 1983, and I'd had intimations about this before, but he reads ahead. They made clear in no uncertain terms about the intellectual superiority of the Birmingham over the Edinburgh School. And while I do have some personal beliefs, I've read the major monographs from each of these, that is Mather and Jenks and Falconer, and but especially given the location of the meeting here, I will not attempt to further adjudicate this issue at this moment. Lindon, however, really was historically the major figure, although I think clearly David Folker needs important credit here, through which the rich intellectual tradition of this Birmingham school entered into, initially in a small amount and now absolutely full bore, into the field of psychiatric genetics. So he really played, as the history of psychiatric genetics is written, and we draw our various pedigrees, Lindon really has to sit there as that main conduit. As I think has been pointed out before, Lindon himself was the cultural great-grandchild of Ronald Fisher, and that David Folker was certainly his overall sibling. Now, I could have told lots of personal stories. I did resist that here, and I think I'll probably use my time in a little bit more serious manner, trying to organize Lindon's contributions to psychiatric genetics. And I thought that there were three major categories within which to place this. One was the issue about data collection methods. The second, and I actually think the most important, it has been emphasized by earlier speakers, is the conceptualization. And the third was a range of these very specific data analytic methods. Again, I will be repeating earlier speakers, but really try to give you a little bit of sense about what the impact of these various issues have been in psychiatric genetics in the 25 years since this task really began. Now, let's kind of transport ourselves back to the real around 1980, and how were we collecting data in psychiatric genetics? What was the methods that we were using? Well, there were certainly a bunch of twin studies. You know, people like Ian R. Kringlin and Eric Essenmuller and Eric Stromgren and Irv Gottesman had gone and collected twin studies of severe disorders. They had the following key methodologic features. First, they only studied rare disorders, all through hospitalization. Vast majority of them were schizophrenia or a manic depressive illness. The results were really presented as concordance rates. So MZ concordance, DZ concordance. Sometimes, if you look hard, they might have measured an environmental variable like birth weight and commented some on the relationship between discordance in birth rate and discordance for schizophrenia. And the typical statistical analyses, if there was any, was a chi-square, looking at whether the MZ concordance rate was greater than the DZ concordance rate. That was really psychiatric twin studies at that time. Now, there were also questionnaire studies being done at the boundary between psychiatry and psychology, which we know is a porous one. For example, Cederloff in the Swedish Twin Registry, so they were analyzing hereditary factors of spontaneous cough. They really learned you could send out questionnaires, large numbers of them, get them back, analyze them. Again, there are people who know this more than I, but the statistics were kind of crude concatenations. Holsinger's H, if you've gone through and looked, they had some basic idea. You had an MZ and a DZ concordance. You had to do something with it, but there was no conceptual framework, no grounding of that in statistical genetic theory. And then also, to give credit, you had a few people that were working with registries. For example, Christensen's study in Denmark of criminality and criminal registration. And again, like the earlier clinical studies, where they actually did go and interview the patients, the end result was concordance rates that were usually simply presented qualitatively, but might have had a chi-square test. Now, there were other things going on in psychiatric genetics at the time. For example, the epidemiological end of psychiatry, Myrna Weissman would be a good characteristic figure. She knew how to ascertain probands for family studies, and family studies were now being extensive. They understood the concepts of comorbidity, that if I begin with probands with major depression, lo and behold, I find higher rates of alcoholism in their relatives. The analyses ended with odds ratios. That was effectively the approach. And Seymour Keddy had done some elegant ascertainment work in adoption studies, but again, the output was chi-square tests. So really, are there more schizophrenics in the biological relatives of schizophrenics than the biological relative controls? So that was the really context. Now, I came to MCV in 1983, and has been noted, I entered in and joined as both the one token American and the token physician, so I played several roles at once, and I learned to drink tea, at least I tried, and we would have these charming conversations that Andrew and Nick and I think to look through about sort of how this emerged, but by the end result was, after I'd been there about 18 months, we wrote an RO1 grant, to try to do what we thought then, and I think it's properly to say, was a new paradigm of a twin study to which we really all contributed in various ways. First, it began with true epidemiologic sampling, because we really wanted to get a representative sample. You don't want to see psychiatric illness through the keyhole of who comes in for treatment, because that produces big biases. We really had to do structured psychiatric interviews. That's something that I knew. How do you really do high-quality assessments? I was not then and am not now satisfied that we can detect the subtleties of psychiatric illness from questionnaire data. Nick and I have had an ongoing discussion of that for many decades about what's the value of those two methods. We might continue that. Third, we really wanted to pay careful attention to environmental risk factors. So you have to look at the instrument that we eventually created, all those precious pages, more of them are devoted to measuring things like stressful life events and parent-child relationships and social support, because we wanted to assess those with equal adequacy. We had to figure out the right sample size, and I have a variety of flashbulb memories of those days together, but one of them was Andrew walking. We had these we were in this small little coterie of rooms, windowless, in the 11th floor of Sanger, and we decided we were going to try to figure out how many twins we actually needed to study, and we decided, well, we think maybe that if we thought that major depression had a 35% heritability, how many twins, let's assume that there won't be any C-score, how many twins would we actually need to study to have 80% power to detect that? And Andrew was walking out and he says, it's 1,000 twin pairs. And we looked at each other and thought, that's absolutely impossible. How could you ever actually get, that's 2,000 people, to get interviews with all these people? So that was the beginning of the process. So you had to do power analyses ahead of time to know how much to study. And of course, 4 years later, we were writing a project for 7,000 twin pairs. So our hubris got high. Particularly importantly was that the intellectual framework for this twin work, for the assessment of the concordance, for the multivariate analyses, for the problems of comorbidity, for the relationship to the environment, would not this kind of ad hoc structure that had been sort of pasted together in this funny gamish that then represented psychiatric genetics, but it had this firm, theoretical, elegant, evolutionary informed framework that really derived from Birmingham through Lindon and applied to humans. So that was the new paradigm, and I'm very pleased to say that this paradigm has now been widely used by many others. And I think if you look through, and if you were to talk to people in the field of psychiatry unrelated to genetics, the sense is that this has had, and the literature deriving from a range of these studies, many that have been with Nick and Andrew and Matt McGew and others, have made it a very important contribution in the things we know about psychiatric illness now, was from that. So conceptual. This is sort of a continuation. It's going to be harder to convey because I think most of us in the audience will find it very difficult to understand what psychiatric genetics research was like in the early 1980s, with the significant exception of a small group of folks then at WashU St. Louis, Ted Wright, Bob Cloninger, D.C. Rao. The idea that you could form precise statistical conceptual models of familial transmission and then test the fit of those models to observe data was absolutely revelatory. I mean, I remember distinctly the first set of conversations I had with Lindon when I visited him in 1982. He was, as many people have pictured him here, sitting at a blackboard, outlining things, explaining. And of course, I'd been struggling with my moderate mathematical abilities, trying to go through papers I couldn't really get to understand tetrachoral correlations and liabilities, and this light bulb slowly turning on about the degree to which you can form these precise hypotheses and test them. I think we take this all for granted now, but I really can assure you that where psychiatric genetics was circa 1980, this was like a bolt of lightning that I think I was very fortunate to be among the first who was able to appreciate at the firmness, intellectual elegance, and eventual power of applying these methods. And what has been clear and what has so enriched my own career is how often important previous issues in the clinical practice and etiology of psychiatric illness is translatable into these kinds of questions. Psychiatrists are wonderful for waving our hands about. We're a delightful cocktail conversation, and we can wax elegant about various aspects of human behavior. Psychoanalysis couldn't be a better example. But when it comes to the bottom of saying, okay, test that. Tell me how you're going to develop an empirical method. How are you going to get the data? How are you going to convince me that this is real? That's much harder to do, and that this vehicle of what Lindon has provided to the field has been one vital bridge. I think that's really summarizing. Okay, this is a couple of quotes from Lindon's paper. So one of the experiences I've had in preparing this talk is I've now gone back and read the literature that I read in the 6 or 8 weeks before I first met Lindon in 1982. And I've been over the papers. I haven't read them again. It's been revelatory, and the degree to which those papers have mapped out both my own career, the career of the field, and things that the field itself has done. And this has been said by the speakers, but Lindon, it's really quite eerie, I have to tell you, this feeling about your ability to broadly lay the parts that then part of our job has been to figure out how do you collect all the data, all these thousands of twin pairs, assess them, and then we've really been doing the normal science, to use the Kuhnian frame, of really filling in those kinds of questions that Lindon has taken his broad brushstroke and painted out. So just a few quotes. Whatever the shortcomings of present data, there is no substitute for a model-fitting approach which tries to predict the findings from multiple biological and non-biological relationships from a consistent and parsimonious theory. The fundamental position is not any desire to see genetical theory vindicated in the behavioral domain, but the conviction that any theory of individual differences must be quantitative to be testable. Again, even in the 1980s, not as bad in psychiatry as in psychology, how often genetics was in this value-driven way. There were the epidemiologists who were thinking about environment, the geneticists. If you pose genetic factors, the capacity of the kind of scientific structure that Lindon articulated to just get beyond that silliness. That is, you model the genes, you model the environment, you see how the data turns out. It cleared the air in a way that is now difficult to fully convey. Elegant and rigorous would be the way that I would characterize it. So data analytic. So directly or indirectly, Lindon was responsible for the introduction of a range of critical methods and models into psychiatric genetic epidemiology. What I'm doing now is giving you a short list. Almost all of these have been mentioned before, but really translating into what we've learned in psychiatry. I've had the experience, I just told you that. All right, so these, if I were to outline 2 papers that were most important for me, these are the 2 that I've been reading over the last week, flying across the Atlantic, training around, and bussing around various places in England. The progressive approach to non-additivity and genotype environment covariance in the analysis of human differences, and even more important in my view, is the 78 paper on model-fitting approaches to the analysis of human behavior. So what I really set myself is just to go through those paradigms and tell you what have we been able to do with them, which have been rich, which have been less rich, why or not why. So para-analyses. I think we've all pointed this out. You can't get a genetic epidemiologic grant funded without doing para-analyses. Intellectually, there were some other bits and pieces of this here, but this has been a critical and central contribution. I told you the story of our first female-female twin pairs. A lot of people have talked about multivariate analyses. It's difficult to exaggerate the impact of this on psychiatric nosology. The field of psychiatry goes through these convulsions every 10 or 15 years where we rewrite our diagnostic manuals, DSM-5, ICD-11. I've been a DSM warrior, so I started on DSM-3R, I've been in the task force of DSM-4, I'm now smack in the middle of the storm of DSM-5. There are now been conferences that have been held on the basis of these kinds of studies, particularly the evidence that we found early on, Lindon and I and our others, Nick and Andrew were both involved, the stunning finding that the genetic correlation between generalized anxiety disorder and major depression is effectively unity. How much impression that had on the field. We've showed shared genetic vulnerabilities across the phobias. So this is, again, a class of disorders that really where the genetic vulnerability types the nosology. That we were unable to find the lack of specific genetic risk for substance abuse. I've had neurobiologists sort of verbally assault me. How could that possibly be true, since we all know that the origins of cannabis dependence must be invariants in the cannabinoid receptor and so forth, and that's just not what the data consistently shows. the evidence for broad genetic risks of internalizing and externalizing disorders. There's a lot on the metastructure now. Bob Kruger has written extensively in that. Our and others' work showing the genetic framework of those kinds of dimensions have been critical. And now we're applying the next generation of these techniques to individual diagnostic criteria, showing, as you might suspect, that the diagnostic criteria themselves are not genetically homogeneous. In fact, they're busted up into a variety of dimensions also. Longitudinal models, again, Lindon has contributed both theoretically and in his work with the adolescent behavioral development model. These can be both very simple and increasingly complex. The early results were quite important, showing, as I think were noted before, that E tends to be for psychiatric disorders, just like for many quantitative psychological traits, mostly occasion-specific, and A is stable. More complex developmental models have had a large impact in child and adolescent psychiatry. It is clear that genetic and environmental risk factors for psychiatric disorders undergo both quantitative and qualitative changes over time. And I think these results have had an important role in demonstrating the highly dynamic nature of genetic influences on psychiatric illness. I interact quite a lot both with other psychiatrists and with more lay audiences where, in the context of using the word gene and psychiatric illness, calls forth a whole set of stereotypes of rigidity, of lack of change, of faithfulness, of inability to treat. And this work, among others, has shown the empirical rubric of showing what a false idea that is, how flexible and changeable genetic vulnerability is actually over time. Another characteristic that only a few talked about with these issues about measurement. Lindon was very sensitive to the idea that so-called genetic findings could be mimicked by problems of measurement. Now, this is an area where psychiatric genetics has only been catching up. Mike's been working extensively in this. I can tell you that at the DSM level, boy, this is only now beginning to make the smallest amount of impact, that you really have to think about item performance, that when you're analyzing these categories, they really are psychometric tools that need to be applied. So this is an area that I would say has not maximized its impact overall on illness. Sex effects, Lindon wrote about it. It's played a somewhat more modest role. Here, probably depression has been the one illness where we've consistently seen qualitative sex effects, and now in a variety of molecular studies of major depression, that is being recognized. GE covariance interaction, these are obviously way too rich for me to describe briefly. This is a paper that Lindon and I wrote, I think, in '86. For a fair amount of the psychiatric community, this was the first time they sort of saw these concepts. I think it's fair to say that well, let's start with GE covariance. This has been proven to be a very robust area. I would point out two broad areas of research that have been replicated multiple times. It is clear that the pathway from genetic risk factors for major depression to the phenotype in part pass through the selection into high-risk environments, including stressful life events, low levels of social support, poor relationship to quality, and equally well, if not stronger, the evidence that genetic risk for externalizing traits also pass through environmental factors, particularly deviant peers, easy drug access, and others. So as we are beginning to look at and get further information of parsing the genetic pathways for the major psychiatric disorders, this GE covariance is front and center, playing important roles. GE interaction. This is sort of the oy vey kind of comment. I'm only going to deal here with a latent variable approach. There actually are some areas of reliable findings. I hopefully in a couple of days when I have a little bit more time, we'll talk some about the single gene, the so-called Caspi phenomenon, but certainly not today. It doesn't often account for large proportions of variance, which, as Lindon and others pointed out, that's what the quantitative literature is. I continue to feel that theoretically, this often is really quite important. It is important to show, in fact, that environments can, or genes can influence susceptibility to a range of environmental factors. It's quite robust. Has the field been very responsible in how we've published and analyzed some of that data? The answer to that is no, but that's not a fault you can lay at Lindon's feet. He did his best to try to reverse the onslaught. So a little bit of speed dating then, as I want to try to close. Of the areas that Lindon has worked in that has probably had the smallest impact, it's probably sibling cooperation and competition models. And I kind of wonder why that is. We've been working some with the social impacts of drug abuse of one twin on the other, and I don't know, but it's worth thinking in whether that has been underutilized. The offspring of twin design, I mean, that began with Walter Nance. I think really it's been Lindon and his close colleague Judy Silberg that have mostly been doing that work. Again, a very powerful potential method to understand the sources of vertical cultural transmission. I think the broader issues of parsing sources of parent offspring transmission, again, it's paid less of an emphasis. in psychiatric genetics in the last 15 or 20 years. Assortative mating, again, less of a focus. I think Hermine and some of us, perhaps partly empirically, when you look in the data that's available, you don't find assortment for psychiatric disorders much higher than about 0.3. And actually, most of the time, that level isn't enough to really wonk out many of the other correlations that it's been so important. Adoption, sibling, and half-sibling designs Lindon wrote about, and I'm pleased to say that now several of us, I think Paul Lichtenstein is here, we've been working with the Sundqvist, as these large Scandinavian population registries have become available, the chances of really working with not only literally millions of sibling pairs, but hundreds of thousands of half-sib pairs have opened up the opportunity to go back to some of these. They were really very difficult designs to implement before, although theoretically having a great deal of power. So, closing comments. What characterizes Lindon's achievements here, to me, are both the breadth and depth of his insights in the field, the prescience of his insights. Toward the end of his 1978 paper, Lindon writes, "What we need now are the data. Theory and method have currently outrun knowledge for want of data." And so I think it's been the role of some of us in the room here, in part to go seek, often with a good deal of effort, to try to collect the samples that would give us power to get at least part way to answering the questions that have been posed by Lindon and colleagues. More personally. So I think Lindon's influence was not entirely a result of his intellect. They were also a result of his personality, and for me, I would close by noting three features. These have been commented by others. First is his scholarliness. We are sometimes in a world of sort of flashy, salesman-like intellectuals. I think we know some of them. Sometimes they go very far in academics. Lindon is the opposite of this. He will provide thoughtful, scholarly comments on Christian theology, if you wish to ask him, some obscure medieval aspect. He has a deep grasp of basic statistical genetics and evolutionary theory. He knows how genes work, and you can count on him to provide a thoughtful comment. Those of you that have been with him in seminar know that he'll usually raise his hand, he'll start with a few apologies, and then he'll say something like, well, I must be being thick here, but, and some people at that point run for the door. If you happen to be the speaker, you usually kind of grab the hold and say, all right, I'm going to hold tight for this coming, usually insightful question. Is there any chance I'm going to get out of here with having been coherent in response to his insightful questions? Others have emphasized this, and it is Lindon's enthusiasm. My first meeting with Lindon in 1982, I think it was typical in several ways. First, Lindon had written me, I never met the man before, and told me that I was to meet at a certain spot in the then very small Richmond airport. Of course, we didn't have the web then. I couldn't look him up and Google him and find what he looked like. And I dutifully arrived on time at the airport, and I went and sat at that spot, and no British-looking gentleman was around to be found. It was a fairly crowded airport. I was looking around. Finally, I gave up, and I had a home phone number, and I called Sue, who I had not met before, and said, could you please describe what your husband looks like? And there I found Lindon kind of meandering up and down the halls of the airport looking for me. Of course, he didn't know what I looked like either. And that was how we first met. But what then happened, Lindon had cleared his schedule, and for the next 2.5 days, I had all this pent-up interest in these fields. I sort of threw a glass darkly. I sort of knew. I'd read Falconer. I could understand, but I never had the time to sit down with an expert, and he was patient and enthusiastic. There is something infectious, sparkling about being with Lindon. You go away from him enthused with new insights. It's a remarkable feature, and I think one of his successes as a teacher and an inspirer is this, and of course it reflects his own approaches, his own excitement and joy in the science that he does. And last, and many again have commented here, so it's his generosity. He is the opposite of the career-focused academic. You keep your eye close, I want to publish high-profile publication. Lindon loves to share. He has the basic excitement, he has an openness of spirit, a sense of being a part of a broader community dedicated to unselfish inquiry to better the human condition. And so I can't think of a better way to praise Lindon than that. Thank you, Lindon.
I'm touched that he finds that amusing where the rest of you don't seem to find it so amusing. So, very good. This is the Lindon is Old Fart ceremony. let's see, I remember accidentally once calling him an emeritus professor rather than a distinguished professor, and he said, that means I'm an old fart. So I think the Festschrift has now maybe undone the sin a little bit. So I'm going to do probably a little more personal than scientific. There will be, scientific bits in there as well, but the first time I met Lindon was 25 years ago now, I guess, and I was 4 at the time, and he, Lindon walks in, and he's a big presence, and he arrives, and I said, you have a very fat tummy, 4-year-old, point right there. And Lindon charmingly replied, you have a very big mouth. And so it was, and so it shall ever be, I think is probably where we are in the context of the community. So, Lindon, it wouldn't be the same without you. I think a lot of people have said similar things to this, but it's, for me, the thing that I find so rewarding when interacting with Lindon is that it's not just in the definition of the methods, and I mean, that's really hard work, and it's really useful, and he's really made great contributions there, but it's also, he really tries to provide intuition about how these methods are meant to work. Is what you are doing making sense? Are you actually, logically following the model in any sort of consistent, sensible fashion? And it's really that kind of intuitive approach to statistics that I find really, really appealing. I find it a much easier way to think about data, to think about the very complex problems that we're trying to challenge in the context of human genetics and psychiatry and so on and so forth. It's that intuitive streak that really helps. Well, that and having the math right is, useful too. So, as Dad said, I took a class when I was an undergraduate, and Lindon gave a couple of lectures, and he gave a lecture on likelihood, and he showed a picture like this, and he said, right, if we do a coin tossing experiment, and we kind of calculate what the likelihood is, and we put out the probabilities given the data, based on the parameter values that we assume, we're going to come across this maximum likelihood. And then he just segues into the, yes, likelihood does what you would think it would do. It gives you the most likely configuration of parameters for the data that you observe. And it was really stunningly clear. When you talk about MX and you talk about model fitting and, Urskog stuff and Lizerl and all that kind of stuff, likelihood is kind of written into the fabric and the DNA of the community and the way in which we approach the problem. And really, it was always kind of one of these abstract statistical concepts, and it was having that very simple, precise explanation that I try and now communicate to other people when I talk about likelihood, which they make me do at the summer school, and other such things, that it's really this way of thinking about likelihood, this way of thinking about statistics that is a really valuable thing from a kind of teacher and mentor point of view. With respect to my interaction with Lindon. So I'm going to talk a little bit about data because I was encouraged to talk about some of the things that I'm doing right now, and really let's talk about sequencing because it's kind of exciting and it's the new stuff. And so there's this big large-scale sequencing project that I'm involved in. We're trying to tackle the big psychiatric disease of autism, terrible disease, high risk to, or high cost to families and to the individuals affected. And really, I think I'll just kind of mention some of the things about the case control data and some of the emerging analyses and how that relates to some of the things Lindon said 40 years ago or so. And there were some trios that we did as well, and I can talk to you about that afterwards. But this isn't about me. This is about, again, that kind of Lindon visionary sort of meme that we've been hearing about all day as well. So I'll let you read the quote, but it's really, really important to remember that this polygenic idea and this, polygenic idea that was very much in fashion in the 50s and 60s and 70s, and Lindon articulates it particularly well in this paper in 1977 that he delivered to the Royal Statistical Society. It's that we're expecting small effects, especially for things that are going to be normally distributed or complicated. And really, I think his work with Ken and Dad and many, many others in this room on psychiatry and behavior have really kind of forced the issue that these traits are no different than something like height. When you say to a person that your height is the influence of many, many small genetic factors in some of your environment, what you eat and how much you exercise and all that kind of stuff, people are like, yeah, okay, I buy that, that makes sense, how tall I am, representation of those sorts of things. But when you segue into behavior, people get a little protective. They kind of withdraw a little bit and they get a little precious about themselves because it really is like the most human of our aspects in a sense. And I think Lindon was always kind of right there at the forefront of those lines, arguing that it is a complex trait like all of the others. The other thing that I think does a good job of explaining the kind of person Lindon is one of the discussants on this paper said that Dr. Eaves shows courage in attempting an analysis, always a good thing, and disarming humility in recognizing the limitations. So Lindon will always give you the good and the bad associated with the models that you're doing, where you can go wrong, what assumptions you're making, and how important those assumptions are, and how that relates to the kinds of conclusions that you're making about your data. And I think those are really, really important lessons for every analyst and statistician and geneticist and psychologist when they're trying to understand the nature of behavior and all of the complex traits and phenotypes that we do look at. So fundamental challenges in autism, much like any other behavioral trait, is that there is extreme polygenicity. We have lots and lots of genetic factors that are involved in this, and I think that's been borne out by the genome-wide association data. Some of the data that I'm going to show here I think also supports that. And when you're looking at the coding sequence, so the sequencing project that I'm talking about here, we're really just zooming in on the genes, because it's the genes that matter more than anything else, I suppose. But there's lots of stuff in the genome. But nevertheless, zooming in on the genes, and there's a huge amount of variability in our genes, just like there's a huge amount of variability in our behavior, in our height, in our weight, in our BMI, et cetera, et cetera, et cetera. And so we can look at a couple of fun little bar charts to explain how complicated and difficult this stuff might actually be at a very real level. So taking the kind of 1,000 cases and 1,000 controls, give or take, you can identify all of the loss of function alleles. So these are mutations that terminate one of the two copies of a gene prematurely. So you no longer transcribe one of the two copies. And you can look at the ones specific to cases, and you can look at the ones specific to controls. Under the model that, autism is a nasty disease, something's clearly gone wrong, it might be a very easy, simple Mendelian scenario that we can narrow down and zoom in on. And it turns out there's really no difference in the case-specific versus the control-specific against what you would expect based on the case-control balance. So this is a really kind of consistent pattern across the data. We do gene-based level tests that Li Liu and Catherine Rader really did the final analysis on, and that's a QQ plot. So this is our expected theoretical quantiles. This is what we observe from testing all of the genes in the human genome in 1,000 cases, 1,000 controls, gene-wise test statistics. And that's pretty flat. There's really no signal there. So there have to be genetic effects. We firmly believe this. These phenotypes are heritable. It's just perhaps they require a little more subtlety to try and access them, or that they are very, very modest and distributed across the genome. So confirming that polygenic. And I could have presented any number of data sets from psychiatric or behavioral phenotypes with similar kinds of QQ plots and similar kinds of results. So prophetic words indeed, Dr. Eaves. So the other thing that I really like about Lindon is that he adheres very, very closely to a few precious basic principles. There are always a kind of internal logic when you're talking to him. Occam's razor is always kind of front and center. Don't try and overcomplicate your model if you don't have to. If you explain the data, you've done a pretty good job. That's not to say that the more complicated things aren't there. It's just that we don't have enough information or enough power or any kind of capacity to make any insights as to whether they're real or not. So why do you care, I think, was always the kind of telegraphed message at the end of those sorts of things. And, this dominance alone is not enough, really talks about some of the discordances between the MZ and DZ correlations for a variety of phenotypes. I think this one focuses on neuroticism more than anything else. But nevertheless, the different kind of amounts of concordance between MZs and DZs. And it made the point that even modest amounts of dominance are sufficient, and modest amounts of kind of low-level interaction are sufficient to explain why you would have a big discrepancy in the correlation between MZs and DZs. So just, a kind of diagenic model where you've got SNPs at one locus and SNPs at another locus, a little bit of instability in the risk or contribution to phenotype between those two loci, and that's going to give you a pretty good whack of dominance in the way the variance is structured with respect to the MZ and DZ. And so what we've done is we've kind of taken some of these ideas and tried to pursue them a little more specifically in autism. And again, you've got that pattern that is clearly recognized as our canonical dominance pattern of a much higher MZ correlation than DZ correlation that would be presumed by the liability threshold model. And we can also pose a kind of genetic question, and this is another thing that Lindon really loves to do, which is let us imagine that we have a system and a model, and what does that model play it out itself with? So let us imagine that we have a cystic fibrosis-like gene model that underpins a 20-fold increase to risk to autism. What would we have expected to have seen based on all of the work that we've done molecular molecularly in autism. And you look at the LOD score, the expectation there is less than 1 for 1,000 SIB pairs. You look at genome-wide association, if we had done more than 10,000 samples, which we haven't for autism, we would even then be just only beginning to get into the range where power was actually accessible. So we go through and look at some novel statistical analysis. This is work primarily conducted by Elaine Lim, also in collaboration with Mark Daly, where what we're looking at is basically the rate of loss of function alleles, so those same kinds of nonsense mutations and loss of function mutations that we think are going to be particularly relevant biologically, and we compare the rate of them of homozygous or compound heterozygous loss of function alleles. So this is wherein we might have 2 different loss of function mutations in the same gene, and that might masquerade as basically a recessive Mendelian form of autism. That's the general conceptual model that we're trying to work in this framework. And what do we see? Well, we see 2 lines of evidence that would support that there may actually be a role for some amounts of non-additivity, but only by kind of zeroing in and focusing on something in a very clear conceptual framework did we have sufficient power and insight to be able to even reveal the suggestion that some of these non-additive influences may be there. So we've got 35 cases with 2 loss of function alleles, 13 controls, and then we also see an overrepresentation of bilineal inheritance from parent to child in the trio sample for loss of function alleles. So that kind of suggests that maybe 5% of the cases that we're looking at are, have some contribution of kind of compound heterozygote or homozygous loss of function. So a little bit of non-additivity. And this kind of echoes a conversation I had with Lindon at one of the at the Egmont workshops, where we were talking about A and additivity and what it means. And he says, remember, Ben, additivity is mush. It's just a big contribution and a conglomeration of all of these small little effects that may be behaving slightly non-additively and so on and so forth. But if you sum them all up, what you end up getting is a reasonable amount of additivity in the scheme of things. So one of the other things that's kind of neat that you can do with this kind of data is look at some of the genes. The gene that we look at in particular that draws our attention is USH2A. So this is an Usher syndrome gene that causes congenital deaf blindness. Whenever you have autism and you're doing the phenotypic measuring part of things, you do a hearing test because a reduction in your ability to hear reduces social communication, seems to have a negative impact on emotional development, and what we have is probably a likely Usher's case, kind of masquerading as an autism case. There may be some additional factors going on. We can't actually rule that out, but certainly if you look at the clinical data, there are severe hearing limitations in this case in contrast to all of the other ones that we're looking at. So phenotype matters too. So Lindon in the workshop. So I think this is a picture that Hermine gave me. If you squint really, really hard, you can kind of see that this is a lambda of a non-centrality parameter over there, and there's like a lambda star and n times lambda n star over lambda n for the rescaling of it. And this over here is a kind of probably 2 distributions of test statistics and talking about that non-centrality parameter. And Lindon has really injected the importance of power into the community, like really focused on how well can we statistically test things, how much information can we extract from the data that we've collected, and how does that relate to our ability to draw conclusions about the nature of variation in the population. And now it's not my responsibility to kind of teach power at the workshop, which is a terribly imposing mantle to try and take over from you. I did have the pleasure of working with you this past year on making some new power slides and trying to do things in R that we hadn't really done before in terms of simulation. But you also recognized and understood that genetics is a data science, and it was going to become more and more of a data science, that large-scale sample collection was going to be essential, effects are going to be modest, and so we're going to have to get bigger and bigger data sets. I'm not sure any of us necessarily would have thought even 5 or 10 years ago how big the data sets are actually going to have to be, but you certainly had the insight that more data meant more power, and more power meant better ability to answer the questions that we're interested in answering in the context of human behavior. So I like to end with something maybe a little more silly than anything else. So what does Google Images think of the good Dr. Eaves? So that's kind of one of the first hits, that picture you've seen a couple of times. You see this picture, it's got the title of godgene.jpg, which I think is absolutely brilliant and couldn't sum it up any better. You continue on and then, oh wait, look, you're hit with someone that has worked with Lindon a great deal, and so kind of so tightly connected are you in recent Google space that Pete and your students come up, even if you're Googling on your name. you continue on and you get a picture of Eros and Psyche, so that's a lovely art. And it's talking about social attitudes and the work you've done on social attitudes and how that relates to some of the things that are going on in the behavior genetics community these days. And then Ken comes up, as you might expect. Ken comes up a lot, usually with the DNA, not always, and with more or less hair on his chin and head, I think, is the way of putting it, if I don't overstep my bounds too much. And then twins, you know, the good twin and the bad twin, I think, is really what the message. You got the smoking and the non-smoking and the discordance. But it's difficult to imagine twins and twin methods without thinking about Lindon at some level. And then you'll be gratified that Galton turns up. So Galton's on the list of pictures that come down. You scroll down a couple of pages, as is Darwin. So I think that these are kind of nice extensions of the historical trend that have that you've taken up, and that's really the lineage that we should perhaps think about, as well as Freud. So you're in with the psychiatrist as well. And last but not least, Sue, of course. So, you know, so both were there. And really, it's been an immense pleasure and honor having the opportunity to learn from you, Lindon. Truly, you are a great teacher, mentor, and friend. And I'd like to say thank you with that.